and we can really have more of a conversation and less of just me lecturing. So, as Amanda said, I am a biology professor and I'm an associate dean for our College of Arts and Sciences. And so today I'm just going to tell you a little bit about my path because it ties into careers in biology, which I know many of you are interested in. And then I'm going to talk about some of the genetics and get you to think about these visual comparisons that we see and try to dive more deeply into the genetics of that and touch on the social construct of race. And then I'm going to end with discussing some of the careers in genetics and biotechnology because they relate to the content that we're talking today and some of the technology that's used to find out about the science behind human genetics. And I also have a slide that includes resources for me or for you all. So I'm happy to share those. And so this is a slide that has some images that kind of illustrate what I do as a biologist, as an educator, and they range from teaching. So there's a picture of me teaching in a big lecture hall, introductory biology class. I do a lot of interacting with local groups like yourself. And so this image is us doing gel electrophoresis with a group of middle school students. And I also have research students that I engage in different projects. And this is a picture of a recent student of mine, Shelby. She's actually gone on to be a biology teacher just this year, got her first job after graduating in the spring. And more recently, as an associate dean, I've been more involved in leadership and helping our faculty and our students have the best experience possible at Northern Kentucky University. So this is an image here at a Latino student event with one of my students, Teresa, and one of our former staff members, Crisia. And then over here on the right, I'm very pregnant in this picture last fall. And I was in Atlanta recruiting faculty members with some of my other colleagues here from Northern Kentucky University. And just to give you all a sense of working at a university is really great because it's kind of like its own little community and we have things like athletics. And so this image here is a picture of my son, Braxton, with Victor E. Viking, an award-winning mascot at one of our basketball games, which I'm looking forward to some of those normal things about life resuming shortly. But I presented this slide to show you that working as a college professor, your days are never the same. There's always different things going on and different ways that you can interact with different groups. And that's really what I love about my job. So how did I get interested in genetics? When I was your age, in the mid-90s, I cannot believe I'm saying that. It makes me feel so old. But around 1994, the Human Genome Project was really getting underway, and there was a lot of information coming out about that. And I was in freshman biology. And whenever we had the opportunity to write about a topic that we were interested in and dive more into the research and what was going on in current everyday times, that's what I was looking into, as I was really interested in the Human Genome Project and genetics. And then in 2000, when I was an undergraduate, that's when they really put out the kind of first official draft of the Human Genome Project. And these two individuals were really involved in that, Craig Venter and Francis Collins, and they were on the cover of Time, and the president was talking about it. And so that just furthered my interest as an undergraduate student. So again, I continued to write papers and do more research in that area. So my path has been probably a bit unusual for an academic in that I've been able to stay in the Northern Kentucky region. I was born in the Florence area, and I went to a small school, Thomas More College. It's now Thomas More University, but it's a liberal arts school, and I played basketball as an undergraduate. So I was very interested not only in the biology program, but also the women's basketball team. And I had a family friend that kind of recruited me to Thomas More and took me under her wing. And I loved the biology department and everything they had to offer as well. And so I stayed relatively close to home for my undergraduate degree. And then I wasn't exactly sure what I wanted to do with my biology degree. I was pretty busy in undergrad with basketball and I was doing well in school, but it wasn't until my senior year that I got involved in research, and we were studying a microorganism that infected cystic fibrosis patients. And that was pretty interesting to me. And I applied to graduate school at the University of Cincinnati, and it worked out. I went there for 5 years. I got a master's in biology, and then I ended up getting an interdisciplinary PhD in genetics education. And I worked with faculty members from across the university in the College of Medicine, as well as in the College of Education and the College of Arts and Sciences Biology Department to complete that. And then I got super lucky and was able to interview for a job at Northern Kentucky University, taking me back across the Ohio River. And I got that job and started as an assistant professor in 2007. So for those of you, if you don't know about being a college professor and kind of what that's like, you first get hired on as an assistant professor, and they give you about 5 or 6 years to kind of prove yourself in some regard. And then you get the opportunity to apply for promotion. And so I got promoted to associate professor in 2013. And then recently in 2019, I was awarded full professor. And then I took on this new role as associate dean about 3 years ago. So I'm in my 4th year as associate dean. So even within this arena, I've been able to kind of take on different roles. And that's one of the great things about being an academic. So I'm an educator. I teach introductory biology and genetics courses. I'm a researcher. I've done research in genetics and biology and STEM education, and I have a couple of papers that I've published displayed here. And more recently, like I was telling you, I've taken on this leadership role as associate dean, and I work a lot with faculty and student development and really try to support others at the university to do the things that make a university so great, engaging students in research and creative activity. In our college, we have the School of the Arts. So part of my job is going to plays and music performances and gallery openings. And so I really get to participate in a lot of different things and support our faculty and students. And I just absolutely love that about my job. So what you're here for today, though, is biology. And so I love being a biologist because for me, I really feel like it relates to so much of my everyday experience, and particularly this year from the coronavirus pandemic that we're experiencing. But it also helps me understand social issues such as race relations in our country. And I genuinely feel that having a better understanding of biology, that the biology that underlies these issues helps me process and function in the world. So for me, being a biology professor is incredibly fulfilling. So I want to kind of give you this caveat. For those of you that are looking at the screen, you can see me. I am a white person, and I grew up in a predominantly white Christian environment. That environment typically does not promote discussions of race. We did discuss race in my household to some degree, although it was pretty rare, but my parents were mostly good role models for being open to different people from different backgrounds. Social justice was something that I became more and more interested in as an adult, and I did not feel comfortable talking about race up until the last several years. I did discuss race and genetics in my courses sometimes, but as I engaged more with friends and colleagues of different races and backgrounds, I became more inclined to have these discussions more publicly. And in that regard, I really appreciate Melody Hobson. She's an executive in the business world. And the quote here that she says about being color brave. So I want to give you a caveat that I may not be the perfect person to give this talk to you today. I don't research race. I don't research skin color. but I do know, I think, enough about it to help facilitate this conversation. And I'm going to do my best because I just think it's an important topic for us to look at. And understanding more, like I said earlier, I do believe that understanding more about biology and genetics can make us better humans. So that's my goal today. So a lot of what I'm going to talk about today was highlighted in a special issue of National Geographic from 2018 titled Black and White. So this is the cover of that issue, and I want you all to take a moment and look at the two girls pictured here. And if anyone is comfortable, you might tell me what comes to mind. And remember, be color brave. It's okay. We're talking about race, and that's how we get better at dealing with these issues. People in the chat. Is there anything in the chat?
I am not seeing anything new in the chat other than from Holly Loudon. She asked a question.
Research opportunities using CRISPR. I hate to disappoint you. I haven't planned on talking about CRISPR today, but that's something that we could set up for the future if you wanted. But CRISPR is not for today. Okay, so the prompt was, when you look at this cover from National Geographic, what comes to mind? What sticks out to you? And it's okay if no one's comfortable answering. We will get more into this.
I would just say, so hey, I'm Christine. Hey, Bethany, nice to see you again.
Hey, Christine.
I'm not
join us.
Thank you. I'm not feeling my best today, so I was going to keep my video off, so I apologize. But when I see this picture, I just see innocence and beauty. But that, that's, I mean, the basics. Very long hair, like, yeah. Is there,
dressed in white? I mean, I think obviously they staged it for that to come to mind, right? That you see. So I'm going to reveal the subheading here, which is that these are twin sisters. And the idea was that you see this image, and like Christine said, you see innocence and beauty, but you also see what you might categorize as someone being a white person and the other one being a Black person. So you might look at that and see that contrast. But they are twin sisters. Now, they're not identical, right? They're fraternal twins originating from two different eggs and sperm from the parents, but they are genetically alike as any two siblings. So in contrast, if you look at this image here of 30 different individuals from the continent of Africa, you might immediately identify all of these individuals as Black, with some variation. Genetically, these individuals, when compared to each other, demonstrate the most diversity we find between humans. So this individual up here in the left-hand corner is from a South African group that speaks Nalu. And this individual in the middle is from East Africa and speaks Hadza. And they are likely more genetically different than if you compared your genetics to any one of these individuals. So I think that's a really challenging concept sometimes for us to get our mind around. And so that's why I want us to kind of delve a little bit deeper into understanding the genetics behind this. So kind of set the stage for helping you see some of the complications that arise when making these assumptions based on the colors of people's skin. We're going to talk about genetic differences between chimpanzees and humans. So here we have this image, chimpanzee and a human boy, and human and chimpanzees have about 99% of their genetic profiles in common. So we're pretty similar genetically. Yet, so one way you might make this comparison is looking at chromosomes. So there are 3 different chromosomes here from a chimp, a human, and a mouse for a comparison. And these are all X chromosomes that are stained using a dye in a laboratory and images taken under a microscope. And you can see that the chimp and the human look very similar, right? But the mouse is quite different. It has its centromere, it's kind of that pinched in point at a different place, and the bands are different colors. So both the human and the chimp on the X chromosome have about over 1,000 different genes, and the banding patterns look really similar because the genes are rather similar. And yet there are a lot of differences. So in that 1% difference, that means there's about 35 million differences in the A, T, C's, and G's, right? You all have probably learned about the building blocks of DNA. And so that 1% difference adds up to about 35 million differences. We have mostly the same genes. There are some differences, but mostly for instance, our immune systems are very similar. We get infected with similar viruses and microbes. However, we know, such as the parasite that causes malaria, Plasmodium falciparum, that can infect our red blood cells, but it cannot infect chimpanzees' red blood cells. There's just a slight difference there that prevents that. We also know that gene activity differs. So if you look at 2 stretches of our DNA, they can be turned on and turned off at different times and or different locations. And so we call this gene expressions or gene activity. So we know that there are definitely differences, but we think about differences between humans, we're talking about 0.01% difference between any 2 humans. So that's about 300,000 individual differences. So there's certainly variation that is present, but we generally have the same genes, just different versions of those genes and some differently expressed genes or different gene activity. Does that make sense to everybody as kind of comparing when we look at chimpanzees versus human to human?
I think that was a really great comparison, very helpful to put it into context.
All right, good. So our species, Homo sapiens, evolved in Africa, and recent fossils found in Morocco suggest that human features began as long as 300,000 years ago. But for probably the next 200,000 years, humans remained in Africa. And they moved to different parts of the continent, and there was some isolation. And evidence suggests that about 60,000 years ago, there was some exodus from Africa. And so that's where you see these arrows that are going all around the world. And that may have been the second exodus, but it appears that that was the wave of humans that gave rise to human populations that we see today. So even though there has been some isolation of humans in geographic locations, there's always been migration and mixing that's prevented these significant differences from occurring. So then why are there these variations in skin color? Does anyone have any idea what is the basis for why we have differences in skin color? Does anybody know anything about that? I know we have several school groups represented here.
Feel free to unmute if you would like to ask the question or answer the question, sorry.
Aha, we have a comment about people adapted to living in different environments. Excellent. I can go, that's a great answer.
I think maybe it's depending on where they are, the sun, how much sun they get, they need to have their skin protected from the sun.
Great, yes. So the general, they were living in different environments. A little bit more specific was that the sun played a role in that. So yeah, we see around the world today that skin color is highly variable. Much of it coordinates with where you lived in terms of the equator. So near the equator, there's lots of sunlight, and darker skin was more useful in shielding against the UV radiation. And then as humans moved closer to the poles, both north and south, the problem was too little sun. And so the paler skin allowed for the more UV to get through that layer of the skin and allow for the production of vitamin D, which is required for healthy bones and some other things within the human body. So we know that it's likely the pressures of the sunlight that were playing a role. So there are a number of genes that are involved in skin color. There are 4 of them that are highlighted in this diagram, and you can see that they exist in animals even before human evolution. If you look later on the right side of the diagram, you can see that different versions of these genes cluster for different skin types. So you can see the darker skin. You see the certain variations in populations in Africa and Asia, as well as regions of Australia, sorry. And then the lighter skin variations, you see the genes towards the bottom of the diagram that are found in Europeans and more South Asians and East Asians. So they're showing you here these genes, and that there have been mutations that have occurred to result in more from a lighter pigmentation to resulting in a darker, as well as from dark to light. So this just gives you some sense of the genes that are involved in that visual that we see so often when we look at people. And humans are rather visual, and so that's part of our categorization is we see very well. And so that's part of our, I think, initial inclination to categorize people that way. There is no scientific basis for race, however. Even though there are these genes that we know of different versions that relate to different skin color, that's a pretty small amount of our genome, and they vary depending upon across the world. So we know that race is what we call a social construct. There was this time period that you all may have learned about in your history classes called the European Enlightenment. And this is where individuals in Europe were really focused on these ideas of natural laws that govern the world and human beings. And in Europe, they were white people. And so they tended to categorize people as white and non-white. And then colonization started to happen, right? They were traveling around the world and they were running into different individuals that looked different than them. Obviously, they were categorizing them as non-white. And particularly with the development of the United States, we started to see laws that started to kind of set some different things in motion regarding race. So slavery existed for centuries before the United States was around, but typically slavery was not hereditary. Boundaries were more fluid. Individuals could move in and out of slavery. But when laws started to be enacted in the United States, such as in Virginia in the mid-1600s, we see that hereditary slavery comes about. And that meant that the status of the mother determined the status of the child. And into the 1700s, additional laws kind of put into place these formal distinctions between people that had different appearances. So we know that this idea of Blacks and slavery was really formed along with the United States, and primarily under the control of white individuals. So you might be thinking, okay, I hear you, Dr. Bowling. We have this variation. We know race is a social construct, but wait a minute. We know about there's ancestry testing. So if we can test, that was weird. Siri was starting to talk to me. So if we know that there's ancestry testing, I'm not sure I understand. Oh, geez, oh, peace. If we know that there's ancestry testing, how are we telling the differences between these different groups, right? And humans do vary in some of their genetics based in ancestry, but it is not based in this distinction that we have in terms of Black and white, or white and non-white. So I wanted to talk a little bit today about how ancestry testing is done, just to give you a sense of that. So ancestry testing is done by looking at what are called single nucleotide polymorphisms. So these are single nucleotide A, C, T, and G changes that vary in 1% or more of the population. So you can see here that most of the chromosomes, when you compare human chromosomes, they're going to be identical, right? But we see that there are these single differences. And when they're present in 1% or more of the population, which is actually, we consider quite frequent, then we call them a single nucleotide polymorphism, or a SNP for short. Okay, we can call them a SNP. And if you look at these SNPs across individuals that have ancestral backgrounds from different parts of the world, you can kind of lump them into groups they called haplotypes. So we know that maybe this haplotype, this C here, G here, G here in the genome, are frequently found in individuals from certain parts of the world. So don't let this terminology scare you. It's just here to common language for scientists to use. So then what we can do is we can take these where we know these SNPs are, these stretches of DNA where we know there's this single nucleotide polymorphism, and we can put them onto a DNA microarray or a DNA chip. So there are these glass slides that have, and DNA is very small, right? So they put these little pieces of DNA all along this glass slide, and they're called probes that contain these sequences for these regions. And they can put 1 million or more of them on a really small glass slide. And then they take fluorescently labeled DNA. So they would take DNA from an individual, they would chop it up, they would fluorescently label it, giving it a tag essentially, like you see in this image here. And they would add that DNA to the glass slide. And where there is a perfect match, the DNA, because of those hydrogen bonds between the different A, T, C's, and G's, they can what we call hybridize or bond together. If there is a mismatch, like you see here in the lower slide, half of the slide, then they don't bond, and the slide gets washed, and this would get washed away. And so there would be no glowing from that particular spot. So then a computer assisted by a laser identifies these glowing spots and can identify the haplotypes that are present. So then you end up with data like I pulled here from 23andMe showing that this individual is 13% Filipino or Austronesian. About 3% Nigerian. So they get that from looking at these single nucleotide polymorphisms. Now, one of the reasons I like to talk about this is because I think sometimes people have this idea that these ancestry testing are looking at like genes that are maybe related to athleticism or related to intelligence or some of these other kind of groupings of genes, and that's simply not the case. They're looking at these single nucleotide morphisms that simply relate back to where these individuals kind of have their ancestry. So I'm hoping that that helps a little bit about understanding that. I also want to say that there are different groups that do these ancestry testings. 23andMe is one you probably heard of, Ancestry.com. There are some others out there. And these individuals are making money off of this endeavor. It's interesting to people. There are some nonprofit, more research-based groups that do this work as well. They tend to share their information and put it out there for anyone to use, while the commercial groups do not. And so that can actually result in some differences. So there's been some interesting things, like this reporter from Seattle did 2 different tests and she came up with very different results. And so she was quite confused by this. And I just want to alert you that the information that they're using, particularly these commercial companies, is information that they've curated. And so we really don't know exactly how accurate it is, and they haven't been sharing it publicly. So it's something to just be aware of. Not that they're doing anything wrong, but I think they're refining the science as they go. And I would, if I were going to do something, I would look to more of those research-based nonprofit groups to do something like that. So that kind of gives you a sense of ancestry testing. The differences between humans is really small, and some of the differences that we're actually looking at really don't mean anything other than some of that variation that's just underlining in our genome that doesn't actually make us that different from one another. So I want to make sure if anybody has any other has any questions about any of those topics I just talked about. I do want to spend a few minutes kind of giving you some ideas about some different careers related to the technology. But any questions about any of the genetics that I just talked about or the technology? Keep thinking, and in the meantime, I'll just give you some ideas about careers. There's a variety of careers in genetics and biotechnology, and they require varying levels of education. If you go on to get a medical degree or a PhD, you can be a clinical or medical geneticist that works with patients. A lot of times they're looking at disease and trying to help with hereditary conditions, but there's also individuals that work in understanding more like human evolution and some of the things I talked about today with different genes involved in things like skin color. There's also what's called a genetic counselor, and that's a master's degree. And these individuals work a lot with patients and other medical professionals to help explain genetic concepts, help people get all the information they need to maybe better understand a hereditary condition in their family. That could be something like cancer, or it could be more of a childhood congenital disease. Genetic laboratory research assistants and lab technicians, they have varying levels of education. You can have a bachelor's degree, maybe go on for a master's for the research assistant, but these individuals typically work in labs and help do the kinds of genetic analysis that I was kind of just talking about. There's also a number of careers in education where you're combining science and education, and like what I'm doing and talk to you today about being a college professor. There's also obviously opportunities in K through 12 and informal education as well. People working in zoos and museums and different outreach nature centers and things like that. So lots of different opportunities if you're interested in these areas. I also have a slide that has websites. So the American Institute of Biological Sciences and the American Society of Human Genetics have 2 really nice websites that give you some ideas about the different careers. There's others out there, but these are kind of 2 of my favorites. And I also have a slide if any of you are interested in some of the different resources I use to talk about race and genetics. The The National Geographic obviously is a great resource. The National Museum of African American History and Culture is a great resource. There's an old PBS show called Race, the Power of an Illusion that still, I think, has some really great things about it, even though it's getting quite old. And then the National Human Genome Research Institute and the Genetic Science Learning Center have some great resources. So anybody have any questions about any of this? So Holly, you teach at Anderson and you all were interested in CRISPR. I feel terrible that I didn't come more prepared today to talk to you about CRISPR, but what interests you all about that? What could I talk about that would help satisfy that interest?
If you'd like to unmute your mic, you can totally do that as well, Holly. That way you don't have to like type out tons of stuff.
Hopefully I didn't just generate a lot of confusion. Hopefully I got somebody interested in some of these topics. Go ahead.
They may be doing class changes. I know that was something that some of the teachers have mentioned, that they would have to do class changes.
Gotcha.
So I don't know if that's taking place right now.
Gotcha. I'm curious if anybody kind of knew that that's about how ancestry testing was done, or if they had other ideas. So Victoria has a question. Do identical twins have the same fingerprint? And the answer to that is actually no. So even though those identical twins have the same genetics, our fingerprints are formed certainly dictated by our genetics, but there's also what we would call environmental influences. So how we are positioned in the womb and how certain proteins that are produced make their way from one cell to another does impact how those fingerprints end up being formed. So even identical twins will not have identical fingerprints because they're not just solely determined by our genetics. Does that make sense, Victoria? Do you have any advice for someone looking to go to college for a biology degree? What to expect? That is a great question. And I think that one of the biggest challenges for many of our students, they may be interested in biology in terms of organisms. Many students come and say, oh, I'm interested in medicine, or I'm interested in animals. And that's great. But what we have to start with a lot of our concepts in biology is we go deep into the cell and we start looking at cellular biology. And we also have students take chemistry classes and learn about the biochemistry, the chemistry that's happening within the cell. And that, I think, is really hard for students sometimes. They really just want to focus on, like, I just want to understand medicine and humans. I don't want to learn about plants. Or I just want to study animals and I don't want to learn about what's going on in an individual cell. But to be a good biologist, you have to have that complete understanding. And so I would just encourage students to be open about, if they love biology, go study biology, but know that there's some building blocks that you have to learn in order to fully understand biology. That's probably, I think, one of the biggest things. I'm going to answer the question about genetics a major focus in an undergraduate biology degree, or are there other major parts of biology as well? Yes. So genetics is just one aspect, although in terms of modern biology, it is definitely the kind of underpinning of all of biology these days. Even individuals that work in ecology and work out in the field and look at water quality, they are using genetics to better understand the microbes that live in the water, the microbes that live in the soil, because now we can take a sample of water or a sample of soil and we can do some genetic analysis that allows us to identify actually very small organisms that are living there that are really hard to identify otherwise. So genetics is by no means everything there is to it, but there is usually every biology program requires a genetics course, a molecular biology course. And certainly you can, like at Northern Kentucky University, we have some other upper level courses that kind of would help you go further into genetics. And so you can study cancer genetics, developmental biology, molecular biology. We have an advanced molecular biology class where you're doing a lot of the biotechnology. Or you can kind of take a different path where you're not studying as much the genetics. Maybe you're focused more on organisms and you want to study more vertebrate organisms and focus on that. So it doesn't necessarily dominate your undergraduate biology degree, but it is many times a solid foundation for that. So there was a question about how the slides are worked. Didn't understand when they talk about deletion. Okay, so when you're talking about the deletions, are you talking about, for instance, I'm trying to remember where I brought that up. Anthony, if you could give me a little bit more information where I can remember where I even used the word deletion. For example, COVID news would be ACE deletion. So you're talking about a specific mutation that is found in some individuals that allows them to either be, I think the ACE deletion allows them to be less susceptible to coronavirus because it cannot effectively infect the cells. I think, if I'm following that correctly. So yeah, so there are an example of this. I'll pull up this slide here, just showing the DNA sequence. And there are different types of mutations that can occur. They randomly occur. They occur in your cells all the time. And actually, when you expose yourself to UV light and get a suntan, that actually is an indication that you're getting too much UV light, and you can actually be, that light can mutate your DNA. Now, most of the time it gets repaired and it doesn't cause any problems, but it can cause, there can be issues that occur with mutations. For instance, like two T's next to one another with UV light can cause what's called a thymine dimer. And then when that DNA, when that cell goes on to replicate, there can be a deletion because of that awkward cellular or molecular bonding that is occurring there. So when our DNA is replicated is really when these mutations can occur. It doesn't get replicated accurately 100% of the time. And so individuals that have this variation, that have this deletion, meaning they're missing either one or a series of nucleotides, that changes the protein that results from that DNA. So you all may be learning about how DNA is transcribed into RNA and then translated into proteins, and those proteins are what do most of the work in our cells. And they are the things that sit on the surfaces of our cells that allow certain molecules in and out of our cells. And an example of that are how viruses get into our cells. They typically have certain proteins that work well with proteins on the surface of our cell, and that allow them into the cell, and then that's where the virus can replicate, and eventually that cell releases the viruses, and then that's how we get more virus growing. So Anthony, did I at least address your question somewhat without correct visuals about coronavirus? Okay, good. Maybe I need to put together, well, I know there's other people that are experts on viruses, so they can probably do a better job with that. I love your questions. How do you think genetic testing could benefit someone? Well, that's a really open question. There's all kinds of different genetic testing, and there's all kinds of instances when it may be helpful. So, for instance, I do have a colleague that was having some medical issues. They weren't huge issues, but she was certainly bothered by them, and she had been to the doctor, and the doctor didn't really know what was going on. He made some recommendations in terms of her diet and things like that, but she really didn't know. And then she did the 23andMe testing just because she was curious, and it was for fun. And it did come back and indicate that she was gluten intolerant or allergic to gluten. And so she had what's called celiac disease. And so that was a real trigger to her. Hey, I'm going to stop eating gluten, found in a lot of our favorite kinds of carbohydrates. And it greatly improved her health when she removed that from her diet. So that's an example of something that was pretty, innocuous. I mean, she was not feeling well and things like that. But once she had that piece of information, it was a game changer for her. You might also benefit from understanding if the cancer that you have is a hereditary type of cancer. So if you are a younger person that is diagnosed with cancer, maybe 40 or younger, depends upon the specific types of cancer, but you might go see a medical geneticist to better understand, do you have a gene that puts you at an increased risk for cancer? And that might inform your decisions moving forward. If you're a woman, that might mean that if it's tied to breast cancer, that you have mammograms more frequently. If it's colon cancer, that might mean that you have colonoscopies more frequently. If you find out that you're negative, then that might mean that it was a random thing that happened. You actually don't have a gene that's making you predisposed to this, and so that would change how you have your medical routine conducted in the next several years of your life. It might inform what you do for your kids, right? So if you did have a gene that you knew put you at an increased risk, you might then extend genetic testing to your adult children and say, hey, I was positive for this. It might be helpful for you to get tested as well, so that you know how to conduct your medical management. So I think genetic testing can be really beneficial for a variety of reasons. Another way that genetic testing is used, and there's a company in our own backyard called AssureX. They specialize in psychological kinds of conditions. So maybe children that have ADHD, or maybe they have anxiety. It's not always children, it could be adults as well. There are certain medicines that work better for certain people based on their genetics. So we have certain genes in our genome that code for proteins that process drugs or anything we ingest into our body. And you know this, how some people react differently to different types of medicines. So what they've done is they've identified, very similar to how we do ancestry testing with the SNPs, they've identified people that react better to certain types of drugs depending upon their genetic profile. So you can talk to your doctor and have them run a genetic test through AssureX, and then AssureX will identify what medicine probably will work better for you than others. When in a traditional medical environment, this has been more trial and error. It's been more like, well, let's try this drug and let's try it at this dosage, and then we'll adjust it. But using genetic testing can sometimes give you a better idea about which drug and how much. That whole area is called pharmacogenomics, and that's really a growing area right now. Great question.
Okay, if nobody has any further questions for Bethany, I want to obviously thank her for coming and doing this wonderful presentation. I feel like I learned a lot about genetics that I didn't know. I'm hoping all of you guys also learned about